Hi everyone, welcome to the session Transcription in Prokaryotes. The key points of the discussion are the components of the transcription machinery in prokaryotes are template DNA and the RNA polymerase, the enzyme, and the mechanism of transcription, which we'll be discussing in 3 steps. Initiation. In initiation, we'll discuss the promoter recognition and abortive initiation, which is followed by elongation and termination, whereby we'll see 2 types of terminations, row dependent and row independent types of termination. The information that is present in the segment of the DNA is made available to the cells through the formation of a molecule of RNA. The synthesis of RNA from a DNA template is called as transcription. The transcription denotes a process in which the information which is encoded in the 4 deoxyribonucleotide letters of DNA is rewritten or transcribed into a similar language composed of 4 ribonucleotide letters of the RNA. So here we will detail the mechanism of transcription in prokaryotes. In the entire section of molecular biology, we are mainly concerned with the central dogma of molecular biology. We started the session with describing the structure of the DNA, which was followed by describing the structure of DNA, followed by discussing the replication of the DNA, and now we are at the level of transcription, where DNA is being transcribed into an RNA. And after that, in the next session, we'll be seeing how the messages in an RNA is translated into a protein, where we'll see the process of translation. So here in central dogma, an important step is missing in this particular diagram, that is a reverse of normal central dogma of molecular biology, and that is the reverse of transcription. At certain times or in certain cases, an RNA is converted to a DNA strand, and this process is called as reverse transcription, which is mediated by the enzyme called as reverse transcriptase. So here in this particular discussion of transcription, we'll be discussing the transcription of an mRNA from a gene or a DNA. So transcription is a process, is a general process whereby a DNA produces an RNA. Generally, there are 3 types of RNA in a cell. They are the mRNA, the rRNA, and the tRNA. The rRNA, which is the ribosomal RNA, and the tRNA, which is the transfer RNA. These two RNA molecules, they provide these 2 RNA molecules, which are our rRNA and the tRNA. They function as rather than informational capacity. These rRNAs and tRNAs, they provide structural support, and also they catalyze chemical reaction in which amino acids are covalently linked to one another, which we'll see in the next session, which is translation. So rRNAs and tRNA, rather they provide structural support, or they act as enzymes, but our mRNA, which we will be discussing in this particular session, they function as informational carriers from DNA into which will be converted into a protein. So mRNA are informational carriers, whereas rRNA and tRNAs, they are mainly structural. They are mainly concerned with structural support, and also they are having enzymatic action. So coming to our discussion of transcription in prokaryotes, we'll begin our discussion with the components of the transcriptional machinery in prokaryotes. So we'll start with the template DNA. The template DNA is also known by another terms. They are also called as antisense strand or the non-coding strand. So we'll discuss these terms and then move on to our main discussion. So here you can see a particular DNA which is being transcribed. So here the terms are given. You can see the template DNA as well as the non-template strand. So here the particular strand of DNA which is shown below is called as the template strand. So as we said, a DNA will have two strands, and these strands, they as we said, they are anti-parallel. One will be in 3 prime 5 prime direction, the other will be in the 5 prime 3 prime direction. So even though there are two strands, during transcription only one strand will be involved in the process of transcription. That means any one strand, and transcription occurs, the process of transcription occurs in the template in the direction which is 3 prime to 5 prime. This can happen in any of the template, either this template or this template wherever the gene is present. So in the case which is shown above, you can see that the template strand is the strand which is below, and you can see the synthesis happens in this particular direction which is 3 prime to 5 prime direction. So here this particular DNA strand where the from which the mRNA is synthesized is called as the template strand, and the complementary strand is called as the non-template strand. So in this case, if I take this as our gene which is going to be transcribed, this particular strand is called as the template and this one is the non-template strand. And they are also, or these particular strands are also given another terminologies, that is they are also called as antisense and sense strand. So we'll discuss why these are called as antisense and sense strand. So first we'll take the case with sense strand. A sense strand is a strand which is similar to a sense RNA, which is nothing but the mRNA. So a particular DNA strand which is having more or less similar sequence as that of the mRNA is usually termed as a sense strand. So here in this case, when a particular gene is being transcribed, you get an mRNA, and this particular this particular blue one is our mRNA, and this particular mRNA will have a sequence which is complementary to this particular region or this particular strand. Because this particular non-template strand is complementary to the template, and when the template strand produces a complementary mRNA, it will be same as that of the non-template strand. And now we can see that this particular non-template strand will have a sequence which is similar to that of the mRNA, and hence our non-template strand is called as a sense strand because it is similar to, which is having a similar sequence to mRNA. And our template strand, because it is complementary to the sense strand, it is termed as an antisense strand. So an antisense strand of DNA is a complementary to the sense strand and is the one that will be acting as a template for mRNA synthesis. And another two terminologies are also used to define these two strands. They are coding strand and a non-coding strand. So a coding strand of a DNA has the same sequence as that of the mRNA and is related to the protein sequence that will make up the genetic code. That means the strand which is having the same sequence as that of the mRNA, that is nothing but our non-template strand or our sense strand, is also called as the coding strand. And the non-complementary of the coding strand is called as the non-coding strand. So coming to the new terminology, so we have defined a template strand and non-template strand. After that, we define the template as an anti-sense strand, and this is the sense strand, because this particular set of sequence, it is having a similar sequence as that of the mRNA. And the next was, this is called as the non-coding strand, and this is called as the coding strand. So coding strand, sense strand, and the non-template strand will be having a sequence or a DNA sequence which is similar to that of the mRNA, and hence they are termed so as sense and coding strand. And the complementary one of the template strand, which is complementary to the mRNA, is called as an antisense or a non-coding strand, or simply as a template strand. So now coming to some other terminologies which is related to transcription. They are upstream and downstream sequences. So here in this diagram you can see. So here you can see a point which is called as a start point. So start point is nothing but the particular sequence from which transcription is going to begin, or transcription is going to start. The particular nucleotide from which transcription begins. So we are going to term the upstream and downstream with the reference to this particular nucleotide from which transcription is going to begin, which is called as the start site or start point. So the particular DNA sequence of the template strand, which is seen after the start point. After the start point, in the sense, to state it clearly, the particular DNA strand which is seen towards the 5' end of the start site, towards the 5' end of the start site, is referred to as the downstream sequence, and the particular DNA sequence which is seen towards the 3' end of the towards the 3' end of the start site is referred to as the upstream sequence. So if we are drawing in a diagrammatic representation, you can take it as from the start point, the sequence towards the left is considered to be upstream, and towards the right is said to be downstream, provided the left side is the 3' end and right side is the 5' end. So it is more accurate to define it in terms of its direction as 5' and 3'. So from the start site, the DNA sequence of the template towards the 5' end is referred to as the downstream, and towards the 3' is referred to as the upstream. So transcription will be moving downstream of the start site, because transcription, as we said earlier, it is from 3' to 5' direction, or this particular direction. From the start site, downstream of the template strand, transcription happens. Now the significance of the upstream sequence is that it is in the upstream DNA sequence you have the important region which is called as the promoter. So promoter usually seen in the upstream of the sequence, and you can see in the upstream sequences will be numbered with a negative sign, and the downstream will be referred or designated with a positive sign. You can see here at the start side, the nucleotide will be referred to as +1. That is the nucleotide which starts from where transcription start. From there it will move to +2, +3, etc. And from here towards the upstream sequence, it will start from here -1, -2, -3, etc. So minus sign refers to upstream sequence, plus sign refers to the downstream sequences. So in prokaryotes, the promoter sequences have two important points which is usually referred to as consensus sequences. Consensus sequence is nothing but this particular nucleotide sequence. For example, if a nucleotide sequence is having a particular sequence like this, if this particular sequence is seen throughout prokaryotes. In every prokaryotic organism, at this particular region of the promoter, if such a sequence is seen, if such a sequence is seen in every prokaryote, such a sequence is usually referred to as consensus sequences, which is seen throughout the phyla or throughout the prokaryotic or throughout the prokaryotes. Or such sequences are usually referred to as consensus sequences. And in prokaryotic promoters, there are two consensus sequences. The two consensus sequences are -10 sequence and -35 sequence. So these are the two consensus sequences of the promoter region that is seen in prokaryotes. -10 refers to its position from the start side, that is, it is 10 nucleotides upstream, the other one is 35 nucleotides upstream. That is why the numbers -10 and -35 stands for. So here this -35 is one of the consensus sequences and -10 is another one. And this -10 sequence is also known by another term. It is also called as a Pribnow box. That is a -10 sequence of -10 consensus sequence of the promoter of prokaryotes is also called as a Pribnow box. And the main function of the promoter is, it is at this promoter region the enzyme RNA polymerase comes and binds. If and only if RNA polymerase binds to the promoter, transcription happens, or transcription occurs from the particular starting point of transcription, or the entire gene will be, or the entire mRNA will be, or the entire gene will be correctly transcribed. So moving back to our discussion. So we discussed the template DNA, the upstream sequence, the promoter region, etc., and -35, -10, and the Pribnow box. Coming to the RNA polymerase is also called as a DNA-dependent RNA polymerase. So from the term itself, it is clear. This polymerase, it takes DNA as the substrate and it is synthesizing an RNA, and hence the enzyme is depending on DNA and producing an RNA. Hence it is called as a DNA dependent RNA polymerase. So this is a multi-subunit enzyme which is having, or the holoenzyme consists of a core polymerase and a sigma factor. The polymerase consists of 4 subunits, 2 alpha subunit, a beta subunit, and a beta dash subunit. The core polymerase, or the core enzyme, is having 3 enzymatic or 3 catalytic sites. One is for the polymerase activity for synthesizing the RNA in the 5' 3' direction. That it is having a polymerase activity for synthesizing, a polymerase activity for catalyzing, or it is a 3', 5' polymerase. 3', 5' denotes the direction of the template. 5', 3' denotes the direction of the mRNA. It is also having, the same enzyme is having the helicase activity, and also it is having the unwinding, which is helicase, and also the rewinding activity. The sigma factor is a part of the holoenzyme. It is also a protein, and the main function of sigma factor is to position the RNA polymerase at the promoter. So our core polymerase, because it is having the catalytic site, it can transcribe any DNA. That means if sigma factor is lacking, it will transcribe any part of the DNA, but it will not be a functional product, or a functional mRNA, or a functional rRNA, or a tRNA will not be produced. To transcribe a particular gene, if a gene is to be transcribed perfectly, the sigma factor is required, because only along with the sigma factor, the core polymerase will be placed at the promoter region. After the process of initiation, the sigma factor leaves the polymerase, and the rest of the polymerization of RNA is done by only the core polymerase. So coming to the mechanism of transcription, as we said earlier, there are 3 steps: initiation, elongation, and termination. So first we are moving on with the initiation step. So initiation is nothing but, or the process of initiation is nothing but the binding of our holoenzyme to the promoter region. When the promoter binds, because when the sigma factor is there, sigma factor, when the core is associated with the sigma, it correctly places the polymerase to the promoter region, and the promoter, it starts unwinding the DNA, unwinding the DNA. So it causes localized unwinding of the template by the polymerase, and you can see a bubble-like structure is produced in the DNA, which is shown in the figure. And in the DNA, that purple is the RNA polymerase. The bubble, I mean, you can see in the DNA, the DNA is being unwound, and that represents a bubble, and that's why it is called as a transcription bubble. And after the process, the polymerization of the ribonucleotides, which is complementary to the template strand, begins. So initiation in prokaryotes is something which is called as an abortive initiation. Abortive initiation is nothing but initiation does not happen in or it is not a one-step process in the case of prokaryotes. The prokaryotic RNA polymerase synthesizes short fragments of RNA. That means it will start or initiate the synthesis producing complementary RNA. It will synthesize. So it will synthesize first 5 nucleotides will be synthesized, and after synthesis of the 5, it will stop, and it will again remove the entire sequence. So what is abortive initiation means? Till the RNA polymerase is competent to produce more than 10 nucleotides, or to polymerize more than 10 nucleotides in a single step. In a single step, if the RNA polymerizes more than 10 nucleotides, then elongation, or then it moves. Then the process transcription moves to the second step called as elongation. Until it synthesizes 10 nucleotides in a step, abortive initiation occurs. That means if it halts at polymerizing 6 nucleotides, again it will remove, start synthesizing. If it is halting at 9, again it will remove the nucleotide it synthesizes. So the synthesis continues till it synthesizes more than 10 nucleotides in a single step, and this particular process is called as abortive initiation. So after abortive initiation, elongation occurs. As I said earlier, sigma factor will be released soon after initiation. So elongation is alone catalyzed by the core enzyme, where a nascent RNA is produced. As the RNA, as a new RNA is being produced, it will be displaced from the polymerase. It will not stay inside the polymerase. It will be displaced from the polymerase. It will not form a complex, and you can see an RNA DNA hybrid only in 5 or 6 nucleotides. You can see an RNA DNA duplex can be seen. The other region of the RNA will be out of the polymerase. So as the polymerase is moving downstream, the region which is upstream will be rewinded by the same RNA polymerase. So this is a diagram that you see here. This is nothing but the two animations which represents the process of transcription. So here you can see in this particular point you can see this is something which is called as an RNA DNA hybrid. So you can see the hybrid, that is RNA DNA complementary, that is the pairing can be seen only for 5 or 6 nucleotides. The rest of the RNA, as you see here, the rest of the RNA is hanging out from the RNA polymerase. And in the above diagram you can see, so this is the DNA. It is entering the polymerase, and after the synthesis of the mRNA is leaving the complex. Here the DNA, after it unwinds the DNA inside the polymerase, it rewinds and the DNA is moving out. So this is the DNA here, this is also the DNA here, and this one is the mRNA which is coming out, the single stranded mRNA which is coming out. So this is the process of elongation that is happening in prokaryotes. So coming to the last step, which is called as termination. As we said earlier, there are two types of termination, row dependent as well as row independent type of termination. So in the first case, the termination is dependent on a protein which is called as a row protein. So this particular protein binds to the mRNA and it moves along the mRNA in the 5' 3' direction and of the polymerase, and where it separates the RNA transcript from the DNA to which it is bound. So it will separate the separates the RNA transcript from the DNA to which is bound, and thus termination occurs. In the other case, which is row independent, the polymerase stops transcription when it reaches some special sequence called as a Terminator sequence. Terminator sequence, what it does, it typically folds into a hairpin loop. So hairpin loop is nothing but this particular Terminator sequence forms a hairpin loop which causes the RNA polymerase to release the RNA completely. Hairpin is nothing but structures like this. When structures like this are produced on the RNA, the RNA polymerase will not move forward. So when this does not move forward, the RNA polymerase senses it as a termination sequence, and the RNA polymerase will be released, and the completed RNA chain will be released without requiring any additional factors. Here there is no requirement of any protein. It is because of the formation of hairpin loops that block the movement of polymerase, and in such cases the termination occurs without the help of a protein. The other one, it is done with the help of the protein which is called as a row protein that releases the RNA from the polymerase. So that's all with the transcription that is happening in the case with prokaryotes. When coming to eukaryotes, it is more complex. The process of transcription is more complex compared to prokaryotes, because there are more factors which are required, more proteins which are required for the process. So with this we complete the process of transcription in the case of prokaryotes.